So I'll be talking about tantalizing dilemma in disease scoring statistics. In fact, disease scoring statistics is a statistic mode of prediction of disease that will be actually talking what is going on there and where we are actually. And the term of disease scoring, which is actually use a statistic in a genomic era to actually understand all this genetic disorder and actually to localize a region in the DNA that's cause disease and that have a lot of applications that I'll refer a few of them here. And also disease scoring statistics allow you actually to understand genetic variation that I'll also be talking a little bit in this talk here. So briefly, I'll give you an overview of what genetic variation is, where it's come from, what are the factors, and how actually we can use machine learning and data sciences to capture genetic variations. And also I'll be talking about those disease scoring statistics, which is a kind of machine learning and artificial intelligence within data sciences approach to actually understand genetic variations. A little bit, I'll touch a little bit of bioinformatics analysis, which is actually a area where actually data science is also being applied in bioinformatics. So this is a little bit of our African young people in my lab here, and my lab is computation, population genetics and genetic epidemiology, which is actually based here at South Africa in Cape Town, University of Cape Town, in the Department of Pathology and Division of Human Genetics. So what is variations? You know, as we can see in this picture here, and you look around you, you see we completely different. And even within our family, we look different. Different skin color, different type of hair, short and tall. And then we may ask the question, what caused these variations? What actually caused this variation? And those variations can be in terms of disease, allergy. You know, we have a different allergy, we have a different disease. We observe people have the same disease, but they follow the same treatment, and someone, one get healed quickly, another one not. And we ask, what is variation? And yet we are still the same species as a human. And we see also this variation in recent COVID, you know, and what this variation is. Other people get progress and other people, they stay asymptomatic. What actually underlines this variation within our species as a human being? So we know the story of migration out of Africa, where a small group of people actually within African continent decide to move from the continent to go elsewhere. Elsewhere, they will be actually facing different environments, diets, and after a while, because they are a small group, they need to survive, they start to interbreed between themselves. And by interbreeding between themselves, they create the kind of genetic similarity in their DNA, and what we call population bottleneck, and what we call also genetic isolations. And that's what we can see today, there's a population undergone population bottleneck, like a population in China, Europeans, here population in South Africa as well, by the Khoisan, who actually undergone genocide. Those are kind of population undergo bottleneck. They become themselves genetically similar. Sorry about these disruptions where I am. Wow. Sorry about these disruptions. And actually with a kind of a mutation that occur every time in our life, and also selection that actually the change due to the adaptation to the new environment based on the movement of the populations. Those are the factors that actually shape our DNA and make our DNA to be different, and definitely our phenotype the way our appearance and looks now make us to be different. So what I want to actually pinpoint about genetic variation here is actually the DNA actually is responsible to what we observe, and what we observe we call the trait and the phenotype. And what's happening to our DNA and what cause variation and what cause those variation is different change that's happened to our DNA. And as I refer here, is kind of mutation that occur often and every time, and is random, and also selection that may due to actually adaptation to lifestyle or to a new environment or to climate change that can affect change. And also, if a populations actually are being separated and genetically isolated for a long time ago, and then they came back again, one of those population came back and interbreed, and they call the so-called admixture. So the example of that is when the European population came back, and we call migration back in Africa, they mix with the remaining African, they create the mixed ancestry. And we have an example of mixed ancestry in America, in most of East Africa, North Africa, you know, in South Africa as well. So those are the variations. But now, we observe those variations. How can we use data sciences? Once we have the DNA, we collect those data, how we can use data science to understand those variations, in terms of maybe drug response, treatment response, and phenotype or disease response differences. So this is an example of where machine learning has been used. It's a kind of a principal component analysis, which is a deep learning approach, where actually we have the data, the DNA data of worldwide populations here. I break it down on different 20 worldwide ethnics, and plus some two country of sickle cell, Tanzania and Cameroon, we can see here. And I try to compare how those individuals within ethnics or country are genetically related based on their DNA. As you know, the DNA is also very big data, you know, very big data of the DNA. And once we have the DNA of our populations, we have to put all those populations together and use unsupervised models, a learning model, to do the classifications. As you can see here in a different color, those are different ethnic groups, the way those individuals within the group, they are clustering together. And on the right side here, you can also see a kind of a Bayesian models of phylogeny that actually can actually tell you the tree generation of those different ethnic groups worldwide as well. So genetic variations is another question we can actually ask, which a lot of people they find is like a taboo answer to ask that question. Is race exist? If we look all those people like this picture here, worldwide picture, the way we looks different. Can we ask genetically race exists? This will be a debatable questions. Other people will say, yes, it exists. We have a white, we have a black, and so on, so on. But genetically, is race exists? So if, for example, you have never seen and have any information about Michael Jackson, and just Michael Jackson happened into our audience right now, and then they ask, I have to ask you questions to say, is this gentleman is a Caucasian or is black? I think I may just look the way he looks. I may say he's a Caucasian, he's a European. Then I may be wrong to say so. I may be absolutely wrong. So what I want to point out here is the descriptions of your skin colors, the type of your hair might not actually allow you to classify individuals in a group. But What's actually can be a good classifier here is your DNA, the data that can be used with a kind of machine learning, kind of deep learning, as I show you in this picture here, to classify individual based on the data, based on the DNA material. If I did have Michael Jackson DNA, I could have said that these persons have maybe, this person have 80% of European or 20% of Africans, or maybe is 100% Africans. You know, so the data can actually allow you to well classify individual, then your appearances and stuff. So now the question is how actually we can capture the genetic variations, specific. How we can capture genetic variations. So now we know the factor that create variations, but the question how we can capture genetic variation. One of the example is to use disease scoring statistics. An example of that is mixed models. You know, you just consider mixed model as you can see here, where in the models you can have in a data you can have a group in your data, a group of sick people, group of also healthy people, and you want actually to use their genetic material to see where actually in their DNA there's a differences. So you can construct a kind of mixed models that we call association model, and other people they call disease scoring statistics. So where we can have a predictors, and we can also have a dependent variable, and we can test, build kind of tests of to find the level of significance of particular predictors, which can be a location in your DNA to be associated with a particular disease by comparing two groups of sick and healthy people based on their DNA material. So this is a little bit of historical where we come from with how DNA has been generated and how the data start to be increasing from here and here, and a lot of consortium has been built over the time to actually generate a lot of data just to understand genetic variation. Everything started on 1917, where actually the DNA sequencing technology started with Sanger sequencing, and that progress till the breakthrough was on 2003, where actually the big consortium on how to worldwidely understand human variation with HapMap project, where they actually genotype sequence massive of DNA around the world and try to understand how worldwide population are clustering, Europeans and African, Chinese and Indians and stuff, how they are clustering. And they use a kind of machine learning approach that I've shown you, principal component analysis, which is a supervised models to actually classify people. And that was with the purpose to understand the variation, the origin of human human species. And there was a lot of progress over the year till now. UK 100,000 genome and other initiatives in China, in Australia, in US. Yet Africa is still not having a big consortiums of actually big data genomic just to understand human variation, so that health care can be progressed in Africa. So this is another breakthrough where we started and a different technology of disease scoring statistics that has been implemented. One of them is genome-wide association studies. The other one, they look on a microbiome genome-wide association study. And today they look on a multi-omics approach, where actually you can have the data, the DNA data, which is at the DNA level, and having the data at the transcriptome level, having the data at the proteomic level, and also have the data, for example, for the other species or pathogen, and have all this data, massive data, integrate them, and find approach of integrating different data layer of different measures. So that is where actually data science is moving on the side of omics to understand multi-omics. And there is a lot of challenge in that regard as well. So this is an example of statistics where we are actually right now in genomic. As you can see here, the reds is where all those technology has been performed, and most of the machine learning and the data sciences approach has been developed. You can see most are in Europe countries, India and stuff like that. So you can see African is blue is only 3%. So that means we are very far away to catch up with the worldwide in understanding human variation and getting into the level of technology and into the level of development of new or novel approach for machine learning and data sciences approaches in the era of omics, genomics, and bioinformatics. So this is an example of effort in this paper over here. We try to understand tuberculosis risk, where actually we did develop a genome-wide association method to actually understand and to find a part of the DNA that actually can predispose people with tuberculosis within South African colored, which is actually a population, a mixed ancestry population in South Africa, and have a long history of mixed ancestries. And that population have a high prevalence of tuberculosis. And yet South Africa is more or less actually clean, or I can say not clean, but compared to other countries where the infection is low. But the question was why the tuberculosis is always occur within these particular populations. Then we actually go to have the data of affected and the data of non-affected to try to understand which part of their DNA can be associated with the tuberculosis. And having that data, because it's a mixed ancestry, we did use machine learning where we actually take some of their ancestors, where we know that population may have ancestry from European, Indian, some Bantu from African and East Asians. And within the South African color data together, we put together when we try to learn from the data to understand. And this box plot here show you in the middle here, you see those are actually mixed ancestry individual and show the different colors that represent ancestry contribution in that particular mixed ancestry individuals. And over down here, you see is again, is a principal component analysis where the green one is the mixed ancestry population, which it's found itself in the convex of its genetic ancestor, which is the European, Africans, the Khoisans, East Asians, and some Indians as well. That actually historically contribute to the mixture of South African color. So now to pinpoint the loci or the regions in that population, South African color, that is associated to tuberculosis, that's how we can plot. We do kind of machine learning, and then we could find a particular regions in the genome that is associated with tuberculosis, and that we can plot this kind of plot, Manhattan plots. And also in data sciences, visualizations of results or of the data is also a big issue. And this plot here, it looks simplistic, it's very simple, but we are plotting the data of each chromosomes, and chromosome contain a lot of locations, millions of locations. And we did test all those millions of locations independently, and then we have to present the data in a plot where actually there is a level of significance in our result. And of course, there is a lot of issue in data science-based approach or machine learning-based approach when you do the kind of association studies. There is a issue of false positive result. There's an issue of a sample size problems. There's other issue like asymptotic bias on mixed models that need to actually be addressed. And also the model may not actually have sufficient parameters as well that can be taken into account. For example, the disease model cannot only be that the disease associated to the genetic. There might be environmental interactions, that can come there. There might also be the microbiome that live in the body of our human body that can also interact to cause variation, to cause the disease and stuff like that. So there's a kind of an issue that need to put in place or to put together so that the questions of variation, the questions of learning from the data to get actually knowledge-based information to be to be well appropriately be addressed. So this is where we go. All these kind of things in the area of genomic is addressed on a single locus, but the test can be done on the regions. We can consider the interactions, or we can also, because the data become more and more big, there's a need to consider the way we need to store those data, the way we need to process it. Today, we need to move on the system of cloud computing, which is more beneficial to handle processing and the storage of more the data, particularly in the era of disease. When we collect the data, we can't fool those data. The more you accumulate the data, the more the model can be more efficient. The more the data you have more data and you learn from more data, the more your prediction get much better. So this is actually, again, another breakthrough of different aspects where we can look on the interaction at the DNA as a network. And we can learn from the network and create a kind of machine learning where actually the tests of risk prediction can be run on a network of each location of the genes in a DNA. And that's increase again the complexity of modeling, statistical modeling or machine learning modeling. This is an example of that where we have a genes, and a gene, we know the gene, a single gene cannot confer a particular trait. They interact. But till now, as we are, a gene can have a lot of location, but we are testing on a single location, not even in the entire genes. Yet the gene also interact. So what can be a machine learning can consider to test a risk of a particular disease or to identify a regions within a genome that can cause the disease within all genes. And also to consider that the gene can interact with others. So it's become more complex. So, and also here I want to show that in our body, the risks that we undergo is also based on lifestyle. And the lifestyle that we are having stress out our microbiome. And then again, our microbiome interact with the host. And all these aspects have a different data. You have it from the microbiome that live, millions of them live in our body. You can extract the metagenomic, proteomic, epigenomic from the hosts. As a human, we can have also genomic, proteomic, transcriptomic, and we can also deal with the nitrogenomic as well. So now the question is how we can integrate all those data just for the sake to understand the risk and to better improve healthcare. And that is actually how machine learning and the data sciences will play a role. And I believe that African, because of diversity that we find in African, differences in environments, this is a great place where actually we can learn more, develop more complex model to address our local problem and leverage now this era of data sciences to improve the healthcare in African populations. So, as I say, because that will need a lot of data, a massive of data, a plan of reproducibility of research need to be taken into the consideration. Cloud and computer need to be taken into consideration. high performance computing need to be taken into consideration. This is just to show a pipeline where you have to have a long-term or lifetime data storage, you know, and to have a kind of mechanism, to have a kind of productive server where you can move the data from live storage to productive server, and also to think of the fair data, how the data can be fair and how that data can be shared with the other patterns, and that we think now what can be the speed, the internet, the way we can share the data so that science can evolve. So those are the kind of considerations, and the data management plan in the data science is a key that need actually to be addressed, and also the reproducibility of data science is actually something that need to be addressed if we African, we need to take into considerations where we are, and we want to move forward in the sciences, to catch up in the sciences, to develop more approach so that we can contribute in science worldwide as well. So prospectively here, machine intelligence for human variation health in African need to be taken into consideration seriously, and also in terms of education, in terms of research, in terms of partnership with our governments. We need to engage ourselves, and we have to also fund a way actually to bridge the gap between sciences and industry. This kind of sort of things, like in this kind of a meeting where we can always also invite industry to partnership with them. And I think in that way, we can move sciences and the data science in Africa in another level. Another consideration here is integrative omics. As I say, today we need to find an approach in this area on a way we can leverage data science to integrate more data of different layers of different species, just for the sake for risk prediction, and also to improve healthcare. So I have to acknowledge African Academy of Sciences, NIH, University of Cape Town, and also TWICE, who actually organized this